All right. So once again, everyone, welcome to our August meeting. Today, we're excited to have Dr. Schaefer, who is a chemical engineer with over 25 years of experience in the FATE transportation and treatment of organic contaminants in water and soil. He received his PhD in chemical and biochemical engineering from Rutgers University. Dr. Schaefer currently is the director of CDM Smith's Research and Testing Laboratory in Bellevue, Washington, overseeing a large number of research projects and treatability studies for federal, municipal, and private sector clients. His recent efforts have focused on fundamental processes related to PFAS migration and FATE in both the subsurface and the wastewater treatment plants, as well as PFAS treatment technologies. So with that, I'll stop sharing, and Dr. Schaefer, I'll let you share and start your presentation.
Okay, fantastic. Thank you. Just give me a second to get going here. All right, so hopefully everyone's seeing the presentation screen.
Yes.
Fantastic. All right, well, thank you for this opportunity to present. I really appreciate it, and hopefully this will be some fun, and we'll have some good conversation. So I do want to talk about PFAS leaching to groundwater, but before I get into it, I do want to note where the funding support for this has come from, and also who my key collaborators are. So this is primarily what I'm going to be talking today is something funded by CERDIP, and we've been, I guess we're about 3+ years or so into this project. We have a little more additional work we're doing, but we have at least enough done, I think, to tell an interesting story. But my collaborators here at CDM Smith were Zoom Nguyen and Sean O'Hare. At Aptum, Dave Lippincott and Greg Livornia, Colorado School of Mines, Professor Chris Higgins and Professor Tisa Langasakari, along with Stephanie Shea, who is a doctoral student, and at Oregon State University, Jennifer Field and Emerson Christie. And they really all deserve a lot of credit for all the hard work they've put into this. So what I'll be talking about today is PFAS leaching through the unsaturated zone at sites at former fire training area sites that have been historically impacted with AFFF, and we're really going to focus on doing a deep dive at one of those sites. And these are and this is an issue that's really particularly challenging for the Department of Defense. They're kind of entering in the remedial investigation phase at a lot of these sites, and it's really important that they get an understanding of the mechanisms and what's driving PFAS leaching, PFAS desorption at these sites, as this can be very important for developing cleanup standards and just overall site management. And I think there's a few important questions. I mean, there's been a lot of things, a lot of studies that have gone on at the bench scale, looking at PFAS migration and transport on saturated columns. But there's really, there still remains a pretty big data gap when it comes to field scale data. And as some of you might know, sometimes what happens at the bench scale and what happens at the field scale don't always match up like we want them to. But some of the key questions that are still hanging out there, I think, is this relationship between what you measure in a collected soil sample, and then at least with respect to these VADOS or unsaturated zone samples, and then what the PFAS concentration actually is in the Porewater. And as we'll talk about a little bit today, that can be a little more complex than for many classes of compounds. There's been a lot of work looking at and talking about PFAS absorption to the air-water interface. PFAS are surface active compounds, they're surfactants, and it certainly makes conceptual sense that could be important when you have, say, an air-water interface in the unsaturated zone. And there's been some really good studies by a number of groups to start to understand this and explain this and quantify this. But when it comes to observing the importance of this in the field and verifying that, hey, at a real impacted site, this is important, there's really still, I think, a lack of some critical and certainly quantitative information on that. And then finally, this whole issue of PFAS mass flux versus mass removal. And you can see the little kind of cartoon figure I have on the right here. So as PFAS slowly leaches over time and mass is removed, how's that functionality going to look like with respect to what the pore water concentrations are, which is directly related to that downwards flux to groundwater. So having some insight on this behavior, again, can be very important when it comes to developing cleanup standards and overall site management. So just talking a little bit about this issue of deriving soil cleanup standards and how maybe this relationship between what you measure in a collected soil sample is maybe different for PFAS than other compounds. Kind of I guess the traditional way of looking at this is what you see in that first equation there. So typically when you collect a soil sample and you measure, say, your contaminant concentration in that, you're measuring really the combination of what's absorbed to the soil and what's in the pore water. And you can then relate that to what the equilibrium, say, water concentration is or pore water concentration is by using kind of your classic KD, linear soil water partitioning type of approach. And that's typically serves as the basis for how soil cleanup standards are derived, and it's based on what's going to be the equilibrium concentration in that water. Now there could be some dilution and mixing factors on top of that, but that's kind of the core equation. When you have PFAS, it becomes a little more complex, and you can see this third term that's present in those brackets there, and that's a term to account for PFAS mass that can absorb at the air-water interface. So when you have a pore water system in the unsaturated zone, you have air-water interfaces, and actually that total air-water interface there can be pretty significant. And you can actually have, especially for more hydrophobic compounds, a pretty significant fraction of your PFAS mass that becomes retained at that interface. So that needs to be taken into account when you're doing this type of mass balance. And it can be very important because ultimately that can mean you have less in the aqueous phase, right? So I think we need to make sure we're developing soil cleanup standards that aren't overly conservative and are really based on the science of how PFAS partitions at the pore scale. So kind of getting into it now, I'm going to be talking about a study location, actually not too far from me here in New Jersey, about an hour or so. It's Joint Base McGuire-Dix-Lakehurst, and where our demonstration or our test site is on the Lakehurst portion of the base. And this location is called Historical Fire Training Area 1 or AFFF Area 16. There was actually no fire training here, but they used this for what's termed monthly finished foam reading tests, and that was basically from the mid 80s to the mid 90s, and that's where they kind of would test the stability of the foam and the air or the water foam mix they would use. They would do all that testing, and as you can imagine, a lot of that foam ended up right on the ground surface. The soils at this site are a fairly permeable, silty sand, and the depth of water is fairly shallow, which is kind of very common here in New Jersey, typically ranging from about 6 to 9 feet below ground surface. And I think for most of our demonstration, the water table was probably around 7 feet or so. So the first thing we did at the site is we went out, and this was in like a 2018, 2019 timeframe. So we were going to the site, nearly 20 years or about 20 years after the last AFFF application. So this was a highly weathered, pretty aged site, but this is probably very typical of what the Department of Defense kind of has in their site profile. So we thought this would be a kind of an appropriate site to look at. So one of the first things we did is we looked at the total PFAS concentrations in soil, and we did some high resolution sampling going downwards as a function of depth. And one of the things we found here, and we see this behavior somewhat at a lot of sites, but maybe not quite this extreme. And what you can see on this concentration is you saw by far the highest concentrations were in the top 6 to 9 inches of soil here. So very very biased to a lot of contamination, very shallow here. And then the contamination was, more or less constant as you went down to the water table. Now we think part of what's driving this very high, 20,000 microgram per kilogram PFAS concentrations in the shallow soil is in large part higher soil natural or organic carbon levels, which was, as you see in the box in the lower right-hand corner, much higher as expected in the very shallow soil than in the deep soil. So there's probably a lot more PFAS that's just absorbed and retained to the soil there. We then kind of took some samples back to the lab, and we kind of divided the soil into a shallow and a deep fraction, shallow being 0 to 3 feet and deep 3 to 8. And we then we homogenized the soil, and we use this for a lot of bench scale testing that I'll talk about in a couple of slides. So as part of that PFAS concentration, in addition to looking at what the total concentration was, we did a lot of detailed analysis, and this was done in Chris Higgins' lab at Colorado School of Mines, where they had the ability to look for not only what we term target analytes, compounds we can quantify, but also what's termed suspect analytes, compounds for which there's not analytical standards, but we can still get an estimate of their mass or the concentration in the sample. And we really wanted to look at the composition of the PFAS in this soil as a function of depth. So I have this very colorful graph here, but I think a couple things are worth noting. So the green bars, that's your perfluorinated sulfonates, things like PFOS, and in fact PFOS was one of the primary PFAS here, certainly in soil, and that's really dominating. So those very high concentrations we see in those near surface samples, that's nearly all PFOS in those samples. But we also see some other things related to precursors. And for those of you who may not be as familiar with PFAS, precursors are other compounds which can more readily transform either biotically or abiotically, and then form the more recalcitrant perfluoroalkyl acids, things like PFOS and PFOA, which don't readily transform. So we see a lot of these precursors present still. So it's been over 20 years since there's been AFFF exposures, and we still see precursors present, and that's consistent with what some other researchers have found in their studies as well. But a couple interesting things with respect to the precursors, you'll see kind of on the around a foot, you'll see some of this dark blue here. Those are fluorotelomere sulfonate precursors, and then they kind of go away, and then kind of further down the soil column is actually you do see some of those fluorotelomere sulfonates actually present. So that can certainly suggest there might be some precursor transformation occurring with depth. The other thing we see a lot of is that kind of orangey colors, and there are a lot of other precursors, electrochemical derived precursors that we see there. So there's still, on a percent basis, there's still a lot of PFAS mass in this soil column in this very aged site that can potentially transform. So we think that's important, and I think that's part of the reason why the Department of Defense and the CERTA program in particular is now investing more research dollars to get a better understanding of how some of these precursors might be slowly transforming at some of these sites. Okay, so I'm going to take a little bit of a digression here and step back from from some of this field data and talk about some of the results of some of the lab testing we did with some of that collected shallow and deep soil. So one of the things we wanted to get a handle on was the PFAS desorption kinetics from soil, since that can typically be a very important thing when you're trying to understand leaching and potential migration of PFAS to the underlying groundwater. So what we found is for the deep soil, that's the soil with the lower organic carbon content and the much lower PFAS concentrations, we basically saw by just setting up a very simple some simple batch experiments with a soil water slurry, and you can kind of see what they look like there in the picture. We found that we had equilibrium within about 48 hours, and that was our shortest time point that we looked at. But for the shallow soil, it was a little bit different. What we found, and you can see on the graph on the left there, is that even over 400 hours, so you're talking several days to a couple weeks, we hadn't reached equilibrium, and that was really surprising. This was a relatively sandy soil. Maybe if this was something that was like, 40% clay, maybe we would expect to see this. So this was a pretty interesting finding, and I think we're still trying to totally understand what's causing this, but it was pretty apparent, and we've seen this behavior in other types of soil now. The other thing that was kind of interesting is we saw more of this kinetically controlled behavior with the shorter chain or less hydrophobic compounds. Think of compounds like PFBS, which is a 4-carbon chain, or PFPS, which is a 5-carbon chain. And we think that the reason for this, and there was actually a great paper looking at PAH desorption from soils, is that for the more hydrophobic compounds, there hasn't, all of the readily desorbable material, or the equilibrium rapidly desorbing material, it's probably still there, because it's very hydrophobic. Whereas for the less hydrophobic compounds, that stuff that can readily desorb is probably long since washed away, and what we're seeing is only what's left in the more kinetically controlled domain. So that's kind of semi-quantitative explanation for why we're seeing that, or why we think we're seeing that here. So once we had a sense of what kind of equilibration times we would need, at least for some of those longer chain compounds, we wanted to actually perform some batch desorption isotherms. So again, based on the equilibration times, at least for the longer chain compounds, we performed this what's termed a sequential batch dilution test. And what this means is you start with those simple soil water slurries, allow them to equilibrate, sample the supernatant water, but then replace all that water with clean water. So essentially, you're diluting the sample, and you just repeat this several times, and what you get is a desorption isotherm. So that's how we generated the data here that you see for both PFOS on your left and A2 fluorotelomere sulfonate on the right. So we kind of collect the data, and kind of going right to left, right, as we dilute the data, as we dilute the soil rather. So what you can see, I think a couple of things to note are important here. One is that these desorption isotherms, at least for the range that we looked at, can be reasonably described by a linear isotherm. However, that linear isotherm does not go through a zero intercept. It goes through something much greater than zero. And why we think this is because there's a fraction of that soil, of that PFAS that's absorbed to the soil, that is not readily desorbed. It does not readily come off. And that can be due to some, kinetic mass transfer limitations. That can be due to some other type of thermodynamic behavior. Some people speculate with PFAS that you can kind of get some weird phase behavior with all the different types of surfactants there. So you could have some PFAS that just does not really want to come off the soil that readily, or at least in a rapid time frame. You'll see in each of those figures, there's a red symbol there, a red open symbol there. I think after the experiment, we let the batch system incubate. I think it was for another 6 months. I think that's the amount of time, and looked again, and then sampled again, and you could see that very little additional mass came off. So we think this is a pretty interesting finding, and certainly has some ramifications if you're trying to develop an appropriate soil cleanup standard. These data here, and again, we've seen this in other types of soils, and other people have seen this in some studies too, suggest that it might be okay in some cases to leave relatively high concentrations of PFAS in the soil without too much worry about the PFAS leaching and migrating down to underlying groundwater. So one thing we thought we would play around with here is, could we predict this fraction of or this mass of PFAS that wasn't readily coming off the soil? And again, we kind of went to the literature, and people have been looking at desorption and adsorption and hysteresis for a lot of other compounds for a long time. So we found this one There's one model that came out and or was published at least in 1998. So we tried applying that to our system, and basically it said that fraction. The authors of this model called it irreversibly sorbed. I really don't like to do that because I don't know or believe that it's truly irreversible, but just slowly coming off, and basically relates that to the fraction of organic carbon in the soil, the maximum concentration the soil was exposed to. Now, this was a bit of a guess for us, but we know how the AFFF stock solutions are typically diluted and what they might be, so we kind of use that as kind of a screening level estimate. And then the only other term in that equation is this hydrophobic area per molecule, which sounds like a really complex term to understand. But this is something for these PFAS compounds that is actually readily calculated by measuring the air-water interfacial tension, and this has been done for these compounds since it's a way of understanding their absorption to the air-water interface. So these data were readily available, and what you can see on the chart on the left there, and again, some of these were pretty courageous approximations we think we made here, but when you look at the measured versus the predicted from the model, we actually get very reasonable and I think almost in every case within an order of magnitude predictions of this. So, perhaps this is something we can use as a tool for other sites, or maybe it is we just got lucky at this site. But this is certainly something we want to look into and see if this is a model that can be more universally applied. So the other thing we wanted to look at the bench scale was this issue of PFAS absorption at the air-water interface, and was there a way? What was the way to quantify or at least estimate this for our system? And again, there's a lot of groups doing this, but the approach we've taken in our group, which is maybe a little bit different than some of the others, is we like to be able to directly via mass balance try to measure this at the concentrations of interest and also in the matrix of interest. And we've published a couple of papers now on this, and we basically have two methods of doing this. Perhaps the more traditional method, and this is for people who are into oceanography and interrogating the surface microlayer. We use something called a Garrett metal screen. So it's just a wire mesh that we just gently place on the surface of the water, and basically the thickness that it interrogates, it can be approximated by the wire thickness, and then we can sample that and say, look at the PFOS concentration in that sample compared to the bulk phase, and what we can get from that is a KAW, and that's just a partition coefficient relating the surface concentration of PFAS to the bulk concentration. Kind of think of that KAW as a KD, but instead of soil being the absorptive phase, the air-water interface is the absorptive phase. The other approach we use is a little more complex. It's basically we generate these films in these small pans there, but the films have a known air-water interfacial area, and by looking at the mass that accumulates in the thin film relative to the bulk, we can make the same calculation essentially and get these air-water interfacial coefficients. So what we wanted to do is actually collect some of the pore water at this site and then use these tests, and for this site we actually used the method on the right there, that film method, but we've gotten very agreeable results between these two methods, which is always encouraging. So I have to show this fancy graph of the data here, just to kind of show how everything fits in. So on the left axis is these air-water interfacial partition coefficients, and according to our models, these are a function of concentration. Although that is a hotly debated statement right now to make. And then we have, and it's also a function of ionic strength, which is very, very accepted. And you can see the dash and the solid line are just the models at two different ionic strengths. The many of the symbols on there, all that at least the two of the triangle symbols and the 1X symbols are experiments we did with some ideal systems. And again, we get, I think, really good agreement between those two methods. But on the far right there is the results we got using the Porewater sample at our Lakehurst site. And you can see it pretty much fell exactly where we thought it was going to be. But perhaps more importantly, we now have a measure of this air-water interfacial partition coefficient for PFOS, as well as some other compounds. So as we're trying to understand this mass balance and what the PFAS is doing, we have a way of estimating that. Okay, so now I've just talked about all this great and very interesting, I'm sure, bench scale information. So how useful is this, right? Now we can put all this together and do some great calculations, but what we wanted to do now is, okay, we have the model, we have all these parameters to essentially tell us what's going to be in the pore water at that site. So now the next test was, okay, let's go back to the field and let's really interrogate this and see if the pore water samples that we're measuring are anywhere close to what we think they would be based on all these data we collected. So going back to the site, and we really started installing this test cell. It's almost 2 years, or about 2 years ago now, in July 2020. So COVID did not stop us from going out and doing this work. And what it basically is a 14 by 14 foot cell here that we put sheet piling around the cell down to near the top of the water table. So roughly 6 feet down there, and that was just to isolate that zone. But I'll talk about this a little bit later. We did some irrigation and flushing experiments to help understand the system. So we wanted to make sure all the water was going downward and not spreading laterally. But perhaps more importantly, we installed, and you can see it in the little orangey colored boxes, 11 porous cup suction lysimeters across the site, and they were installed at various depths from about 1/2 foot down to about 5 feet below ground surface. We also installed 3 moisture probes. Those are the blue diamonds there. We had a shallow monitoring well to sample the shallow groundwater, and we also had a rain gauge present. So this was our system, and this is what we used to kind of interrogate what was happening in the pore water. Just a little bit more about the lysimeters. We purchased them from a company called Soil Moisture. The figure on the left there shows how we installed this, and we just used a hand auger for all of this since it was sandy and we weren't going down that deep. But basically, you want to put some fine silica flour at the bottom there, and that's just so you maintain a wetted contact between that ceramic porous cup and the soil. We like putting a few inches of like a playground sand on top of that, and then we like to bentonite up to the surface just to ensure we have no short circuiting or anything else going on. What I have shown here is collecting the samples with a little hand pump, and typically we would pull a vacuum somewhere around 60 centibar or 60 kilopascal, and we would have to wait several hours to overnight actually to get a nice maybe 100 mils of sample in our collection. In our collection vessel. And then on the right, you can just see kind of a test lysimeter we had installed. All right, so here's a picture of our installed field system. You can see obviously the sheet piling sticking up. You can see various all the lysimeters. In the upper right-hand corner, you can actually see the actual lysimeters that we put in, and they were of various lengths based on the depth to which we set them. I think one of the best things we did here, and I have to credit my colleagues at Aptim for helping with this design, is rather than have to worry about going out and with the hand pumps to collect these 11 lysimeters, we hooked everything up to a vacuum manifold system. So that's why you see a lot of those lines running out there, and that little Connex box in the kind of the background on the left there. That's where we had our control system. We could see the cooler that we used to serve so we could hook up those vacuum lines to little bottles, which served as the knockout pots for the sample. So we were able to control the vacuum remotely. So if we had a rainy day, say the day before, we're like, hey, I think today's a good time to collect a pore water sample. We would remotely turn the vacuum on, let it pull away overnight, and have someone come out to the site the next morning and then retrieve the samples. So that worked very, very well for us at this site. Okay, so finally getting into some of the field pore water results. The first thing we did for a month or so was just under ambient conditions, so we weren't running the irrigation system or anything like that. Fortunately, we did not have a very dry summer like we're having this summer. This might be a very much less interesting presentation. As we looked, we wanted to see over 3 sampling events and looking at the shallow and the deep lysimeters. So there were 4 deep and 4 shallow lysimeters that we used. We want to get just to get a sense of the repeatability among samples. So, a couple things here. One is we generally, and we've seen this now at 3 or 4 other sites, we generally get pretty good repeatability both spatially and temporally. And by pretty good, I don't mean plus minus 10%. I think staying within a factor of 2 is probably a reasonable expectation for these types of installed lysimeters. The other thing that was interesting and maybe a little unique at this site is we didn't see any significant difference in generally in concentration between the PFAS concentrations of the deep and shallow lysimeters, and I think that's because at this site we've kind of reached a pseudo equilibrium. The leachings occurring from the most shallow soils and those top 6 to 9 inches, and we pretty much have have equilibrium going down to the water table as things slowly dissolve. That's our explanation for this observation. I guess the other thing is just stepping back and looking at these pore water concentrations, and we've looked at pore water at 4, 5, 6 sites now, and they basically stay within an order of magnitude or so of what we're seeing here. So it could be at a lot of these sites where it's been 20 plus years since there's been AFFF exposure, which is probably a lot of the fire training areas in the Department of Defense portfolio. This might be kind of a typical concentrations, if you will. All right, so how about our predictions? So now we have these pore water concentrations. How well are we or were we able to predict that? So probably one of the most interesting ones is PFOS. That's a compound that's more surface active than at least a lot of the other compounds that we're looking at, and certainly one of significant regulatory importance as well. So the hatched blue on the left is what we got from our lysimeters, and then the red bar in the middle is the prediction we have on the model, and the model is basically what I have shown on the right, and it's just a simple mass balance, right? Looking at what's in each of those phases, and then those KD and KAW terms that we got from the interfacial tension and those desorption isotherms. If we neglect what's at the air-water interface, what we found for PFOS is we had about a factor of 4 over prediction about what was in the water. And quite frankly, even with that over prediction, that's not really too bad. A lot of times, even if you're well within an order of magnitude with a model, that's usually fairly respectable. But what's interesting is that when we do include that air-water interfacial tension term, that accumulation at the air-water interface, we actually get a model prediction that's really within the error of both the field and the model prediction. So we get a much improved prediction there, and that was certainly a nice line of evidence, and I think a strong indicator that this air-water interfacial retention really is important and is having an impact on what we're measuring in pore water and ultimately what's impacting underlying groundwater. So the last thing at this site that I'll talk about is we did this kind of ambient testing. The other thing we did was we wanted to look at, well, what happens if we try to accelerate and wash out what's in there? Can we get some sense of how behavior is going to change over time? So we started using our irrigation system, and we stopped and started a few times to get some intermittent numbers. But essentially, we flushed the equivalent of 886 inches of rain, which is somewhere around 20 years of rainfall that we did in slightly under a year here. You can see on the right, those are results from the moisture probes, and basically what that tells us is kind of what we already knew, is that the water was moving through this kind of sandy, unsaturated zone relatively quickly, and we could basically have pretty aggressive flushing and not worry about a lot of ponding or things like that. So here are just a couple of the results for two of the compounds, PFOS and PFOA. The symbols in red are what we measured under ambient conditions, and for some of the earlier conditions, I kind of rather than putting several symbols, I just kind of took some average there. But what we see, I think two things are interesting. One is overall, we do see as we flush, we do see concentrations in the pore water decreasing, and so at least that's moving in the right direction. The other thing we saw that's kind of interesting is when we initially start the flushing, seems to be a lot more scatter in the data than we typically see, and maybe that's because there's some new water flow paths forming, but maybe it has to do with some other things as well. So we kind of found that interesting. So the last slide I'll show for this site here is just some of these curves we derived that plot the fractional decrease in pore water concentration as a function of the total mass removed. And let's just focus for now on the graph in the upper left-hand corner, and maybe just focus on PFOS. And what this tells us is that we see a pretty large decrease in the pore water concentration, 75, 80% there, with only a very small, less than 10% of the mass being removed. And this is very much in agreement with what we saw with those desorption isotherms, right, which were nearly these horizontal line for PFOS that showed we were seeing very big decrease with a very large decrease in the aqueous concentration, a very small decrease in the soil concentration, and then we would see less so for some of the other compounds. So this is really a nice line of evidence that kind of supports some of this bench scale desorption or leaching data that says, yeah, you could see some very big decreases in pore water concentrations and also mass flux to your underlying groundwater, with only removing a very small amount of mass to your system. Now, for the perfluorinated carboxylates, which you see in the lower right-hand corner, while the overall kind of trend looks the same there, you'll see we're getting fractions of mass removed that are much greater than 1 for a couple of these compounds. And there's really two potential reasons for this. One is we did a pretty bad job estimating our amount of PFAS mass for these compounds in our test cell. And while I can't completely rule that out, we certainly didn't seem to do that poorly for the perfluorinated sulfonates. The other reason is that we could be getting formation of some of these compounds due to transformation of those precursors that we showed in those early graphs. And in fact, when we did some of the batch experiments for our desorption testing, we had much more PFHXA desorbed than what was initially in the soil. So maybe this is an indicator, or perhaps this suggests that we're getting some transformation of these precursors in situ, and we're continuing to kind of look at that. So I just want to wrap up now. I just want to show a couple of quick slides. We have another project ongoing where we're going out to a number of sites, and we're not doing this big deep dive of investigation that we did here. We're just putting in a few adjacent lysimeters and taking a few rounds of sampling and doing some bench scale testing. So just a couple of things, highlights from this work is we looked at the nature of the PFAS in some of these pore waters. So this is Peterson Air Force Base, Air Force Academy, and Randolph Air Force Base, and we see that the nature of the PFAS that we're seeing in the pore water is very different. In some cases, it's mostly those perfluoroalkyl acids, things like PFOS and PFOA, and very few precursors. While as at some of the other sites, we're seeing a lot of precursors in the pore water, including some that are the suspect precursors that commercial labs can't even detect. They have yet to be picked up by academic labs. And I think this is important only because I think it demonstrates that some of these precursors, again, even at these very aged sites, they're migrating. They're potentially making their way down to groundwater, although a couple of these sites have it's a pretty substantial depth to groundwater. But I think this is a pretty interesting finding and suggests again that these precursors can be very important. We also, what we did here is here's some lysimeter data, not dissimilar from what we collected at the Lakehurst site, but we wanted to collect this, compare this to what we measured in just a kind of a batch slurry test. A lot of groups are trying to develop or verify some sort of batch desorption leachate type test to see if we can predict pore water concentrations. And I guess what I'm showing here is with few exceptions, those batch slurry experiments are really not giving us a very good prediction of what we're seeing in pore water. At the very least, you would have to determine a lot of those modeling parameters that would go into that to relate that to what we're seeing in pore water. And then finally, the last slide I show you, I promise, is we're doing these what I term these microlysimeter tests. So we'll take a core, an intact core, back to the lab, and we'll have a very small lysimeter, very similar to what we use in the field, but just a much smaller scale, as you see there. And we use it to pull out the pore water. And when we do that, do we get decent agreement with what we see in the field? And so far, I'd say the answer to that is yes, particularly for the longer chain compounds. For some of the really shorter chain compounds, we're still in some cases nearly an order of magnitude off, and we're trying to figure out why. But this might be, I guess, an alternative way to investigate what's happening in pore water rather than relying on some lysimeters, which at some sites can be pretty problematic. So just to wrap up, some of this kinetically controlled desorption might be really important for a lot of these PFAS sites. We have now both lab and field data to suggest that air-water interfacial retention can be important. And we were able to get a reasonable prediction of PFAS pore water concentrations, at least at this site. We're trying to see if we can repeat this at other types of sites. And then, some simple batch tests to kind of be predictive of leaching and pore water concentrations may not be the best approach. So I know I threw a lot in there, especially if you're not one who's super well versed in PFAS, but I really thank you for your time, and I'm happy to take any questions.
Great. Thank you so much, Dr. Schaefer. I clearly did not know about PFAS, which is why in my introduction to you, I said PFAS, but now I know more.
No worries.
But thank you so much for your presentation. All right, everyone, as Dr. Schaefer said, we're now open to questions. So if you want to put anything in the chat, feel free to do that. Or if you want to turn on your video and your mic, we're also happy to do that. Or Dimitri, I feel like you have a question. You always have questions straight out the back.
Of course. Okay, so I'll start. So Dr. Schaefer, excellent presentation, and wealth of information that you shared with us, both on the kind of testing part of it, as well as sharing data. One graph that really hit me was, I think it was slide 5 or 6, one of the initial ones that you had with depth, where it showed very high concentrations on the surface. Yeah, this one here.
Yeah.
So when you first got those data, to me, it's a bit surprising to see, having heard of PFAS, usually it prefers the water phase. They're mobile contaminants in general. At least that's the sense that I have. So when you're talking about the site that was, I guess, around 20 years old, you mentioned, so undisturbed for 20 years, and in an area that receives a reasonable amount of rain, or are we talking about dry?
No, this is in the heart of New Jersey. We get about 40 inches or so of rain a year.
So was this surprising to you to see such high concentrations on the sediment itself at the top layer, nonetheless?
Yeah, it was actually, and thanks for bringing that up again. So we've gone to like I said, 4 or 5 other sites, and some are in more arid regions, but generally at these sites, we'll see kind of peak of PFAS concentration, somewhere in that 2 to 5 foot range, right? And then we'll also see kind of a bit of an elution profile with kind of more of your hydrophobic compounds, maybe showing the peaks a little more shallower, and then your less hydrophobic compounds, maybe showing another foot or two of migration. But it is a little surprising to see, these compounds aren't just rapidly flushing out of the vadose zone, and I think there's a couple reasons for that. One is they are very soluble in water, but these compounds will sorb, and especially if there's organic carbon and things like that. So they're not behaving by any stretch like tracers. The other thing, and this is an active area of research, is there's some speculation that especially starting from time zero on these things. I mean, there is a whole mix of these AFFF formulations. There's hydrocarbon surfactants, there's fluorinated surfactants, there's other things. People suspect there might be some weird phase behavior going on, and some hemimicelles forming, or some other aggregate type structures, and they've certainly been observed in some other situations. So it could be that, at least for some period of time, these types of things were serving as ways to retain these compounds in the subsurface. And I think people are still scratching their heads a little bit on that. And then the other thing, at least for some of the longer chain compounds, you do have this air-water interfacial retention, right? Which can, as we saw, can be a factor of 3 or 4, or maybe even more for some of these compounds. But all that being said, this site, at least for the handful that we've looked at so far, appears to be a little more extreme and a little more unique than some of the others.
I see. Well, thank you very much.
I'm still absorbing all of the information. I feel like my mind has been blown. All right, looks like we have a question in the chat. It says, "Thank you, great presentation, Dr. Schaefer. Was there any other characteristics besides organic carbon content, perhaps related to soil surface property, linked to absorption to soil particles in your studies?"
No, excellent question. And we looked at just a couple of things. We looked at anion exchange capacity and cation exchange capacity, and it's really the anion exchange. Most of these PFAS that we're looking at here are anionic. But when we sent it out to the lab, the anion exchange capacity was, essentially 0 or negative according to their numbers. So I don't know. That certainly didn't give us much insight. We did do some work on looking at the clay content, and certainly even that little amount of clay, especially in the deeper zone, might also be responsible for some of the sorption. But we haven't looked at too many things beyond that. But there's certainly a lot of groups who are looking in much more detail about the soil properties and how that relates to. Most of the studies are adsorption, probably still a little less unknown with desorption. But that's an excellent question.
Thank you. Looks like that's everything in the chat, but if anybody has any other
nobody
questions.
else has a question, I have a couple more.
Sure.
Actually.
Go for it.
So, I'm going to stay on this slide, actually.
Okay.
I know it was outside your scope to check for, bioaccumulation or anything in microorganisms that are residing in that top layer where you see these high concentrations. So, based on the research that you've done and the papers that you've read on PFAS in general, what type of concentrations would you say that that you would find, and let's say in lipids, in organisms that are residing in the top layer of the soil?
Yeah, that's a great question, and that's probably one I can't give you a good answer on. There's that's kind of one area that I have kind of certainly don't have, well, one of many areas I don't have expertise on, but one that I haven't really done a deep dive on PFAS. I know there's a lot of good people doing good work on that, But I mean, but maybe to your overall point, especially for this site with such high concentrations on the surface, I would think there would be potentially a much greater, kind of bioaccumulation, biotoxicity potential impact here than at some other sites, and it would be interesting. Again, it's really not what we're doing here, but since you mentioned that, I should perhaps provide this information to some of the folks that are doing this, because there might be a lot of interest in looking at a site like this.
I would definitely agree. And sorry for putting you on the spot with regarding biological.
Not at all. It's a great question.
So another question that I would have here. So this site was undisturbed for 20 years. Based again on your knowledge on PFAS, how do you think would be over those 20 years those surface concentrations would evolve to reach this point here? I mean, how much higher do you expect they were like 2 years or 3 years after they were released?
Yeah, so that's another excellent question, and probably still an active area of research. And here's the real complicating factor. So when these AFFFs are released, there's a lot of co-solvent there. There's a lot of hydrocarbon surfactants that are in fact readily biodegradable. When that AFFF's released, it affects the surface tension of the water because you have such high concentrations. So now that affects how water's percolating down there. So there are a lot of complexities, and I don't think it is well understood. During those initial, the initial time the soil is exposed to AFFF, what happens now? Now I know there is a research project, and I think Mark Brousseau at Arizona and Bogu are involved with it. They are going to perform that type of experiments on a little plot and get fresh soil, expose it to AFFF, and then see what happens. So I'm very interested in seeing how that looks like. That's the front end. I'm kind of looking at the tail end and seeing what's happening, but there's a lot of mystery in between there. So I think you're spot on with your comment.
A lot of questions to ask with these sites. Yeah, a lot of insights.
Yep. Yeah.
Didn't know if you had another one. I was going to wait for you.
No, I have another one. I'm waiting to see if anybody else is going to ask. I have another question on the next slide, actually. The one that had the distribution, the composition.
All the colors.
Yes. So on this one here, you have, if I remember, the precursors on the orange one. So, of course, since you have much higher concentrations, you have a lower percentage of precursors on the top, top high half a foot. Mm-hmm. But their absolute concentrations would be much higher than deeper, obviously, right?
Yeah.
So still, you have those precursors at the top layer, higher concentrations, and you can see them, historically moving downwards as you go down. Thermodynamically thinking, I don't know how that transformation happens from precursor to the actual compounds. I always think that it has to be thermodynamically favored. But do you have any sense on how that process happens over time? Is it just stochastic and it happens under any conditions, or it's more favorable at deeper, less sunlight, let's say, or less water?
Right. So you're asking all the right questions, and that's where the reason. In fact, CERTIP just had a call and just made selections, and I think are funding about 7 projects, including one or two of ours, to answer some of these questions. How are these compounds transforming? What are the rates? What are the expected end products? So there's been a lot of bench scale studies that have shown various biotic pathways for this. A lot of these can co-metabolic processes could be really important, especially in the very shallow zone. But there have been studies at least to show some direct metabolism, at least of some of the precursors, right, to form under anaerobic conditions to form other compounds. We've shown that some abiotic processes can be responsible. Things like ferrous minerals, right, and even some of the other low levels of hydroxyl radical that could be generated with shifting redox conditions. I'm sure, at least for some of the compounds directly on the surface, there could be some other, UV types of transformation occurring. So the nature of what's transforming these compounds, right, and maybe more importantly, the rates at which they're being transformed are the questions that we really need to answer to understand what's happening long term. And we suspect that early on there's a lot of compounds that are readily degradable, right? Be them precursors or those hydrocarbons. So kind of what happens in the first several days, maybe the first couple of years in transformation might be a lot different than what's happening 20 years later. And I imagine what's left 20 years later is a little more recalcitrant, right? Because of bioavailability or other factors. But that's what we need to understand. I mean, I think what we've proven here, I think what we've shown here is that a lot of these precursors are still around. We have good evidence that there has been a precursor transformation. I think the question is to what extent and what rate is precursor transformation occurring now at a site like this?
Excellent questions. Yes, because there are so many sites all over the country like this at different stages as well.
Yep.
Excellent. Thank you very much for everything. For all the information and the questions.
Thank you for all those good questions, man.
Yeah, thanks, Dimitri.
You're good at this.
All right. Well, last call for any questions in the chat, or if you want to speak up. We have the last few minutes in our. Oh, here's another one that came through in the chat. It says, "Thank you for the information. I think it's supposed to be informative presentation. Did you measure any difference in pore water concentration in various depths in monitoring wells?"
So not for this project, but there's a project we were on with. So do you mean actually within the borehole of the monitoring well, or just at the site at different depths at the site? Maybe I can clarify that question a little bit. Well, let me try to answer it both ways. So, there has been some concern that because of some of the surface active properties of PFAS, that even within a well borehole or casing, that there could be some stratification occurring. And I think we've shown, and Jennifer Field put together some paper on this that doesn't really occur. There's just not enough area and the geometry. But in terms of in terms of this site, is there much of a did we see much of a concentration gradient with depth at the site within the saturated zone? I would suspect there is, but that's not something we really looked at as part of this study. We really just focused on the unsaturated zone here.
Great, thank you. Well, we're about at the top of the hour, so thank you, Dr. Schaefer, for your presentation. Thank you, everyone, for your questions, for attending. Just as a reminder, our next presentation is on September 15th. We'll have Derek Marks from the Tulalip tribes talking about helicopter large wood placement on the Tolt River. So we look forward to seeing you all there.
Looking forward to.
Yeah, thank you again, Dr. Schaefer. Really
thank you all very much for your time. I appreciate it.
Thank you very much, sir. Yes.
Have a good day, everyone.
Have a nice day.
Thank you.